Okay, so I did not record the lecture in class because we had a student on Zoom who, yeah, anyway, situations. So I'm doing it now after class in my office. So if you have questions, jot them down, email them to me, or let's talk in class. So I started class by going over this question. This was the question that most people got wrong on the quiz. So I wanted to give you guys another chance to review this material. So what I want you to do is for your attendance for today, which is March 31st, what I want you to do is tell me what's happening in each of these 3 stages in PCR. What's happening during the denaturing stage? What's happening during the annealing stage? What's happening during the extension phase? Feel free to refer to your notes, Google stuff, do whatever you need to do in order to get this information into your brain, because you probably are highly likely going to see this on the exam. So this is not the last time you're going to be asked questions about PCR. Okay, so make sure you understand what's going on there. That's that. There's a portal on Canvas where you can turn this in. So just write me, somewhere between 2 to 6 sentences about what's going on. So a couple of sentences for each of these steps. Okay, so we are ready to start talking about cell reproduction. And today we're going to focus on asexual reproduction. So cloning, mitosis, so building. So just like we started out as a single cell zygote, and we had to make copies and copies and copies of ourselves to get into our multicellular form. So we're going to talk about that process. It's called mitosis. But first of all, let's talk about prokaryotes. And let's remember that prokaryotes are single-celled organisms. They are very simple compared to eukaryotes. They have one circular chromosome that's held within the nucleoid region. That's here. This little squiggly line is one chromosome that's circular, that's bunched up into a little ball like that. And sometimes we see plasmids in the cytoplasm. And if you remember from when we did our gene cloning, we made this little bracelet thingy, that a plasmid is a circular bit of DNA that is separate from the actual chromosome, that's copied by itself. And so that's what a plasmid is. And so what happens is that these bacteria and other prokaryotes reproduce through cloning in a process we call binary fission. So first thing that they have to do is order to replicate their DNA, because if we need some DNA in this cell, we need some DNA in this cell. So we got to make a copy of that DNA, and that's what's being shown here in this figure. All right. And so then the next thing that happens is that there's a bunch of proteins that are called the temperature filamenting temperature sensitive mutancy. You don't need to know the name of that protein. Okay, the proteins tend to collect in this middle, the center of the cell that's starting to elongate. Half of so with this replicated genome, we see one chromosome kind of go over on this side and one chromosome go over on that side. And then these proteins kind of form this ring where we start to see this, it's starting to pinch off into, instead of being this one like oblong cell, it's starting to pinch off to create 2 cells. And so that's what's happening now, is that we have the cytoplasm is starting to be split in between these 2. We see the septum, it's the plasma membrane, and the cell wall are going to eventually pinch off, boop. And then we are going to have 2 daughter cells that are genetically the same as each other and as the parent cell, assuming that DNA replication didn't introduce any mutations. Okay, so prokaryotes can only reproduce through cloning. However, they can introduce some genetic variation through this thing called horizontal gene transfer. And we're taking that plasmid and we can swap it with another individual. So there's three main ways that horizontal gene transfer can occur. And it can be through transformation, just like we did with this. And we can actually incorporate a foreign bit of DNA into a plasmid. And now we have a bigger plasmid, or we might actually have some of that be removed through splicing. And so then that would be a different set of genes happening in there. We often see conjugation. So we have two individual bacteria that are going to come and fuse, and they're going to share cytoplasm, and that's what we're seeing here. And so now the plasmids can transport back and forth. So we're actually seeing these two bacteria, their plasma membranes fuse, and they can cell organelles and plasmids and cell juice and all that stuff. And then they break apart and then go their own ways. We also see horizontal gene transfer through this process of transduction, where a virus will actually come onto a host bacterium, pick up a bit of its DNA, and then transfer it to another individual bacterium. So those are the three ways. Lists are my favorite thing because I can just throw a couple, like one or two wrong answers in there, and boom, there's a multiple choice question. All right. So now let's talk about this thing called exponential growth. We often see that bacteria will undergo exponential growth. And in fact, all populations do this, including human populations. We have this nice J curve. I learned this stuff from a Canadian, and so he called it a hockey stick curve. But let's go through this. And I forgot about this. So let me grab a whiteboard real quick. So what we can do is we're going to say that there's a bacterium that is going to divide every 20 minutes. And so what we want to know is to get a number of how many cells are going to be produced after 2 hours. So the first thing that we want to do is figure out how many times that cell is going to reproduce. And so that's 120 minutes in 2 hours, right? And so if it's doubling every 20 minutes, then that means that there's 6 doublings. So I'm going to just write that down here on my whiteboard, 6 doublings. And so we're going to start with one bacterium, and it's going to make 2. And then each of those is going to make 2. Now we could keep drawing this out 6 times, or we could do math. So we're going to double. So that's going to be this part. And now our exponent is the number. So where n is the number of doublings. Yeah, that's crap. Okay. So in this case, we're going to have 2 to the 6th power, which is another way of writing 2 times 2 times 2 times 2 times 2. Okay. And if we put that into our math machine, we're going to get that we're going to have 64 bacterial cells after 2 hours if they double every 20 minutes. Okay, so make sure that you understand how to do that, how you could draw that out, or how you can math it. And if you forget how to do the math, you can definitely draw a little diagram in the margins of your paper. Okay, so make sure you know how to do that. So now, that was all the cell replication prokaryotes that we're going to talk about. The rest of what we're going to talk about is for eukaryotes. Now, remember that the structure of eukaryotes is a lot more complicated. They have a nuclear envelope that their DNA lives in. We've got multiple linear chromosomes, and the DNA is wrapped around proteins called histones to make these things called nucleosomes. And oh man, it's so much more complicated. And so that's why we're going to get into all of this terminology so that we can use this terminology to explain what's happening in eukaryotic organisms. So the first thing that we need to talk about is ploidy, which is just a fancy science talk for saying how many numbers of each chromosome are there in the nucleus. And we can write it shorthand by saying X is, some number, whatever number we're talking about, and N, in this case, is going to be the number of chromosomes in there. And so if we are talking about a haploid critter, like fungi usually are haploid, they have one copy of each chromosome. And we can either write that as 1N, or if we're feeling lazy, we can just write it as N. It means the same thing. Us multicellular animals, we tend to be diploid, where we have 2 copies of each chromosome. So we're going to write that as 2N. Okay. So now you'll see that in a diploid organism, for this chromosome number 1, there's 2 chromosomes. For chromosome number 2, there's 2 chromosomes. For chromosome number 3, there's 3 chromosomes. And so we're going to now use this term called homologous chromosomes to describe those 2 copies. And so these are chromosomes that are the same length, and they have the same genes in the same location. When we're talking about where genes are located on the chromosome, we call that a locus. Okay, because it's location, so it's a locus. So we get 1 set of our chromosomes from mom, 1 set from dad, and those chromosomes might contain different alleles. Now, an allele is gene alternative. So if we have, so I asked in class, I said, okay, somebody give me an example of, some trait. And so they said height. And so if we, I've got a lot of shorty genes. So I have all the alleles for being short. But my oldest kid is almost 6 feet tall. So like they got some of the tall genes or some of the tall alleles in the family. They didn't come from me, that's for sure. All right, so that's what that is. This over here is the image of the chromosomes of a human. And so we call this image a karyotype. And so again, karyo meaning nucleus. So we can actually see these different chromosomes and tell if there's too many chromosomes, not enough chromosomes, misshapen chromosomes, shortened chromosomes. All those things are going to tell us a little bit about how, because usually what we see when there's issues with the number of chromosomes or the shape of them is that these people tend to have a lot of health problems. And so if we know, oh, okay, this kid has an extra 20 or a third 21st chromosome. So instead of having 2 21 chromosomes, they have 3. Okay, so now we're going to say, okay, that kid has Down syndrome. This is how we treat Down syndrome. So it's a nice, useful diagnostic tool. Okay, let me go back to this, this slide that I skipped over really quickly. So we don't need to know life cycles for this class. That's something that you'll spend a lot of time on when you get to 182. But I did want to just point out, this is why this helps us describe, gives us a little bit of context of why we talk about haploid and diploid. And we're going to talk about it more later on. But so if we have for animals, oops. Oh, what did I do? Okay, we have our multicellular life stage as a diploid. We have specialized cells that are going to undergo a process called meiosis, we'll talk about next week. Those are going to create haploid gametes. So we just get rid of half of our chromosomes. And then when sperm and egg come together to form a zygote through fertilization, now we're back to being diploid because we get 1 from the sperm, 1 set of chromosomes from the sperm, 1 set from the eggs, boom, now we're diploid again. Plants and algae do this weird thing where they have a multicellular diploid individual and they have multicellular haploid individuals. Worry about that when you get to 182, but this is, I just wanted to give you the context of why we're talking about ploidy. Okay. So we've already talked a lot about DNA compaction, but this is going to become important when we are starting to talk about the stages of mitosis. So I just wanted to remind you that when I would talk about the chromosomes condensing, this is what we mean. So instead of just being a double helix that's just kind of floating in the cell, they start to pack it up tight so that these chromosomes become visible under the microscope, and that allows the genetic material to be put in the right places so that the resulting cells from these cell divisions are going to end up with the the right chromosomes. Okay, so now we're ready to start talking about the cell cycle. All right, and this is section 10.2 of your textbook. So the cell cycle looks like this. So there's two major phases. There's interphase, where the cell's just kind of like hanging out doing its cell thing. And then there's the mitotic phase, where it's actually actively dividing. And so if we look at this pie chart, as kind of think of it as a clock, what we see is that the majority of the time of a cell's life is spent in interphase. And so interphase is all of this gray area. And then just a little bit of it is of the cell's life is spent in the mitotic phase. So so let's just say that this baby cell has just been born. We have to go through a period of growth. And so we call that G1 because this is growth cycle number 1. So we're going to be lazy and call it G1. And then once it has acquired enough nutrients and energy, it can start to synthesize DNA. And so this stage of interphase we call, where their DNA is being copied, we call it the S phase because S stands for synthesis. And so after that DNA has now been copied, oh, that was really tiring. We need a period of recovery. So we have a second growth period. We call it G2 because we're lazy. This is our G2, our second growth phase before, and we're acquiring energy before we go through this highly energy intensive period of mitosis. Okay, and then the mitosis is kind of split into two phases, like real disease mitosis and this other thing called cytokinesis that's kind of happening at the same time. And so we'll talk about that. Okay, so this is our, so let's break this down and let's talk about what's happening in each of these phases. So in our growth 1 phase, or our first gap, or growth 1, however you want to call it, cells are just doing their cell thing. They're just making proteins, doing some cellular respiration. And when we look at them under the microscope, it doesn't really look like anything's happening. We can't really see, like maybe we'll be able to see the nuclei, but it doesn't look like much is going on. They're acquiring building blocks for DNA synthesis, so getting those nucleotides ready. They're making proteins, and they're doing whatever the job of the cell is. Okay. So now we're going to move in. So there's some kind of signal from the environment that says, all right, buddies, let's make some DNA. And so now we enter the synthesis of DNA phase, the S phase. The DNA is still in its chromatin form, but now it's being duplicated. And what that's going to do is give us two identical chunks of DNA that we call sister chromatids that are attached at the centromere. So here's a chromosome. Here is the homologous chromosome, maybe one from mom, one from dad. And then when it duplicates, now we have a chromosome that's made up of one sister chromatid and another sister chromatid. And the homologous chromosome is made up of one sister chromatid and another sister chromatid. Those sister chromatids are attached at the centromere. That's right here. So this part is confusing for a lot of students. So make sure that you understand that this is a chromosome and this is a chromosome. This chromosome is made up of two sister chromatids. I know. Why couldn't we give this a different name? I didn't name this stuff. I found it just as confusing as you did. So here we are. That's what we're stuck with. Okay, so after we have created this two sister chromatid chromosome business, now the cell's tired, and that was a lot of energy that it expended. So now it needs to have like a little rest period, time to refuel and get ready for the next bit. We might actually see some of the We might see some more ribosomes being created, maybe some more Golgi apparatus, and so we're going to see some duplication of some of these organelles that we talked about. And we also got to get rid of that cytoskeleton. If we're too rigid, when we try to pull apart to make a new cell, we're not going to be able to. So we actually need to remove some of that infrastructure of the cytoskeleton. Okay, so that's all happening in this second growth phase, in the G2 phase, or second gap phase, however you want to call it. Okay, so now we can start talking about the mitotic phase. This is where the action is really happening. And even though the cell spends only a little bit of time in this, we're going to spend a lot of time talking about it because it's complicated. So basically what's happening here is that those duplicated chromosomes, keep bumping the computer and not meaning to. So we've got our two chromosomes. We got our chromosome made up of our two chromatids attached at the centromere. They're going to be aligned, separated, and move into two cells. So mitotic phase has really broken up into two phases, where we have karyokinesis, where we actually have the movement of the chromosomes. So carry, right, like we, that's that means nucleus. Prokaryote to eukaryote, right? So karyokinesis is the movement of the nuclear stuff. And then cytokinesis, cyto means cell. And so we have cell movement. We're actually going to be splitting the two, the cytoplasm into two, roughly half for each new daughter cell. All right. Okay, so I already talked about that. So what we end up with after mitosis is two genetically identical daughter cells. When we talk about meiosis next week, we're going to see that that's different. So mitosis is two genetically identical daughter cells. Let me move my, okay, yeah, so there we go. So in reality, mitosis is a continual process. So we have this nice gradient of things happening. And so we're going to break that gradient down into stages. And so what we're really going to see is that there's a little bit of arbitrary and there's a little bit of overlap in each of these stages. So we're going to have to just make some judgment calls, and we're going to have to decide where we're going to put that. That cell could be, you could either call it prophase or you could call it metaphase. And so you have to decide which bucket you're going to put it in. Okay. So we definitely have, there's like 4 to 5 stages depending on, your book uses pro-metaphase. I tend to ignore this one because I think it's really just more prophase, but I'm a lumper. I like big categories of things instead of lots of little categories. So, but anyway, so we got prophase, pro-metaphase, metaphase, anaphase, and teleophase, and cytokinesis, which is a different process, but it's happening simultaneously. So let me show you this hand movement that one of my students taught me several years ago, and I was like, that's brilliant, I'm keeping it forever. So we can talk about prophase, and then we'll go to metaphase, and then anaphase, and then teleophase. And that'll make more sense as we go through these. So keep those prophase, metaphase, anaphase, teleophase hand movements in mind. And as you're taking your quiz or your exam, I'm going to go, which one of these stages is that thing? Okay, cool. All right. So let's talk about prophase first. What's happening in here? Well, first of all, we got to get rid of the nuclear envelope. We got to free up those chromosomes so that they can move around in the cell. We see the chromosomes are starting to condense and starting to become visible with our microscope. We also see in animal cells these spindle fibers starting to form. And they're going to be kind of on opposite ends of the cell. And so if we looked at the onion roots, which we did in lab, and you guys will do, this is what prophase looks like under the microscope. You can see that this area of the nucleus is getting really condensed. It's stained really, really dark red right there. We're starting to be able to see the chromosomes individually. Okay. And then, so this is kind of in between prophase and metaphase. So if we have prophase, metaphase, and this one's kind of like somewhere like this, what's happening is those chromosomes continue to condense. They become more visible. We see the cytochrome, sorry, I looked up how to pronounce this word and I don't remember how to do it. But anyway, it's a bunch of proteins that help move stuff around. The centromeres are going to keep moving toward the poles. So basically in prophase and prometaphase is basically more prophase. It's just getting more extreme. So if you're going to forget any of these phases, forget prometaphase because it's kind of like somewhere in between prophase and metaphase. All right. So again, let's just talk about this. So we have one chromosome here made up of two sister chromatids held together by the centromere. The kyanochore, I think is how you say it, but don't quote me on that. That's going to be proteins that attach to the centromeres that allow the spindle microtubules to attach to so that they can pull those two sister chromatids apart. Okay, so now metaphase. This is my favorite phase because it's so easy to see when we look at it under the microscope. So what's happening here in metaphase? Our mitotic spindle is complete. So here we have that mitotic spindle. We have this structure going from the centromeres to the spindle fibers. The chromosomes are lining up along the equatorial plane, so down the middle of the cell. And so that's why hands are like this, because it's lining up those chromosomes. And then we see that the chromosomes lie on either side. So as they get pulled apart, they're going to end up in the proper cell. So if they were aligned this way, then and they're pulled apart this way, then we're not going to get the right chromosomes in the right places. So the alignment of those chromosomes is really important. Okay, and then we have the spindle fibers attaching to each of the sister chromatids. Okay, so we did prophase, we did metaphase, now we're in anaphase. And so what's happening now is that those sister chromatids are being pulled apart by those spindle fibers. And now, instead of talking about sister chromatids, each of these are now, this is a new chromosome, this is a new chromosome. So what we talked about when they were in metaphase, they were sister chromatids, but poof, now, bless you, my son, you are a chromosome. Okay, this is what this looks like under the microscope. Okay, so we did prophase, metaphase, anaphase. Now we're going to get to telophase. So what's happening here is that our chromosomes have arrived at the poles of the new cells. We're going to start building back our nuclear envelope, and we're going to get rid of those mitotic spindles. We don't need those anymore. And then this, and then in cytokinesis, the cytoplasm is split in about half-sies. So for each of the new daughter cells. In animal cells, we see this cleavage furrow as the new cells start to pinch off. In plants, we see a cell plate, which I think is happening here. Okay, so here's just a nice little summary of what these stages look like under a microscope, at least in plants. This misspelling was not me this time. But I think this is a useful figure even with a misspelling. So here in this plant cell, we can see the cell plate starting to form here between the two daughter nuclei to make these into two new cells. Okay, so yeah, so I kind of already talked about this. In animals, we have the cleavage furrow that's pinching off the cytoplasm, and by pulling the plasma membrane down until it pinches off into two. And if you kind of think of balloons, how you might twist a balloon, like one of those long skinny balloons, that's kind of what's happening. And then the cell plate is growing in between the two daughter nuclei and eventually become the cell wall that those two cells now share. Okay, Sometimes we see a G0 phase, which is where the cells are not actively preparing to divide. So remember, G1 means active preparation for division. G2 is we're like really getting ready now. But in G0 or G0, sometimes we call this, the cells are just kind of, they're quiescent, which means that they're inactive. They're not preparing for replication. And it could be due to a lack of nutrients. So there's just not enough energy in order to fuel reproduction. Or we might not be having the external signals that say, no, it's time to reproduce. And then we also have cells in our mammal bodies that never or very rarely will divide. So some examples of that are our cardiac muscle cells. Those stick with us our whole lives. They never divide. That's the same cell that's in your heart was there when you were born. Some of your neural synapses, your neurons never divide. And so those are kind of a few examples. So, and of course our skin cells, they're constantly dividing because we're getting shedding our skin cells all the time. And so we need to replace that. Same with our esophageal and our intestines. We need to replace those cells all the time. But our brain cells and our heart cells, you know, those are us, really. Okay. So now, we can talk about asexual reproduction in eukaryotes. So remember when we talked about prokaryotes, they only reproduce through binary fission, which is asexual reproduction. Prokaryotes can't do sex. You have to have nuclei in order to do sex, and then they don't have them. So, but eukaryotes can do, some eukaryotes can do both sexual and asexual reproduction. So what are some of the methods? So usually single cellular organisms can do binary fission, just like prokaryotes can, where we actually are copying the DNA and then just splitting the cell juice. And now we have two daughter cells that are the same as the one parent cell. And genetically the same. And we're just splitting the cell juice and replicating the DNA. We can also see budding in some of these eukaryotes. So a good example is plants that asexually reproduce through rhizomes. So aspens do this. If you know creosote bush that's down in the Sonoran Desert and Phoenix area, those are huge, 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 huge clones that are tens of thousands of years old that reproduce through these underground stems. And so what happens is this plant will send out these stems that live underground, and then after it reaches far enough away, then it'll shoot up a new shoot. And if I came through and with a shovel and I cut this right here, this little individual could still continue to grow. It's genetically the same as its parent, but it can live independently of its parent. We can't pull off our thumbs, boop, and then grow a new me, right? So we can't do this. But there are some animals that can. So for example, there's this little sea creature called a hydra. They're like this big, they're cute as hell. And they have these tentacles that filter feed out of the water. And what they can do is that they'll just grow a certain growth. And then eventually when it's big enough, it will fall off of the parent and it will land somewhere and it will continue to grow and live independently of its parents. Genetically the same, but now there's two individuals. Baker's yeast does this, and you can see them here. Here's one yeast cell giving rise to another yeast cell. Okay, and then we can have fragmentation. So one of the ways that you take advantage of this, one of the reasons why cottonwoods are such a great model organism is that they clone really easy. And so I can go walk around with some big old pole pruners and cut down some branches of some cottonwood trees, take them back to the greenhouse, put some rooting hormone on them, stick them in some soil, and now I can have thousands and thousands and thousands and thousands and thousands of trees. And I know that they're genetically distinct from each other, or genetically the same. If they came from the same tree, they're genetically the same. And so now when I do my experiments, I can hold the gene component steady, and I can change the environmental conditions in which I'm growing those trees. And now I can start to get at that nature versus nurture question by holding the gene steady, by taking clones of those same individuals. It's pretty fancy. So there's my friend Allie back there, and we planted all of these cottonwood trees out at a restoration site on the Little Colorado River. Some animals can do this. So we have a flatworm, a little image of a flatworm here. You can cut it up, and then each of these fragments will grow into another individual. These are all genetically the same as each other and genetically the same as the parent. And so that's a pretty cool little thing to do. And I've actually seen these flatworms in the wild for reals with my own eyeballs in the Colorado River. It was pretty cool. I was like, oh my God, I thought these only lived in biology labs. But it was cool to see them in the wild. All right, so that's fragmentation. This is just a nice little summary slide for you that shows animals doing asexual reproduction. Okay, and then the other thing that I wanted to point out to you guys is that for lab 10, what we did is first for activity 3 is that we looked at some microscope slides of some fish, little babies, little embryos, and we were looking to see if we could find the different stages of mitosis. So here you can kind of see one that's doing anaphase. You can see the spindle fibers pulling those chromosomes apart. Here's one in prophase. So here's one in metaphase right here. So I put some pictures up on the assignment for in module 12. If you look for that lab 10 assignment, you click on it. There's the link to a Google Docs that has some of these images. So this is for activity 3, you're going to look at the fish. For activity 4, you're going to count a bajillion cells. And so here, look at this nice anaphase right there. That's beautiful. That's textbook. Here's a nice metaphase. Here's a nice teleophase. Here's a nice interphase. So what you're going to do is you're going to count up the cells in each of those stages and kind of get an estimate of how much time each of those cells is spending in each of those stages. Okay, so that's on there. I'll show you real quick before let's see, I got to get out of this. Okay, so now if I go to, oh, I got to sign back in. So if I take us to the canvas, oh boy, this is not where I want to be. Okay, canvas, here we go. Take us to our course. You can go to modules. We're going to find module 12. We're going to find lab 10. And then if you click on this lab 10 photos, even if you weren't able to attend class in person today, you can still do this lab activity. And so here's a field of view. Here's a field of view. Here's a field of view. And then just for completeness, activities 1 and 2 you guys can do on your own. They're basically worksheets to help you understand the process of mitosis. So you should have everything that we did in class today available to you now. And let me know if you have any questions. I'm available via email on Canvas, and then so I'll just try to make sure I'm watching those. And I will see you guys on Tuesday